However, since microtubules take into consideration many important functions in cell resting and differentiation, including cell division and intracellular transportation, antimicrotubular drugs demonstrate their disadvantage by creating a variety of unpleasant symptoms. For example, immunosuppression, serious fringe neuropathies, myelosuppression, and gastrointestinal toxicity. Natural products ranging from penicillin derived from the penicillium notatum mold to remarkable chemotherapy tranquilized Taxol derived from the specific nutrient enhancement in medicines. Several microtubule binding polymerized microtubules. Many studies reveal that paclitaxel boosts the elongation and nucleation of the microtubule. The tubulin convoluted electron crystal structure show that the beta subunit has a binding site for paclitaxel, and paclitaxel arrives at its binding site via tiny openings on the microtubule surface or using dynamic microtubule fluctuations. Paclitaxel's attachment to the microtubule's binding site tends to stabilize the microtubule lattice, hence polymerization is enhanced. Apopolones are a class of macrolide drug microtubular stabilizers. Apopolones A and B are a member of a unique group of antimicrotubule drugs and are naturally synthesized by Myxobacterium cerangum cellulosum. Primarily for cytotoxic and antifungal activity, they were isolated, which was conjoined with the polymerization of microtubules and meiotic arrest. Both apopolone A and B give off an impression of being contending with paclitaxel for its coupling site, yet their remarkable molecular arrangement ties a location near the taxane binding site. With reduced toxicity and enhanced stability, several synthetic and semi-synthetic compounds have progressed. It has been shown that a natural apopolone B, known as patubulone, is 20 times more effective than paclitaxel. It has appeared to penetrate the blood-brain barrier effectively and has a toxic profile restricted to ostensibly neurotoxic diarrhea. In comparison to paclitaxel, ixabebolone, an apopolone B derivative, has 5 times more cytotoxicity and is uniformly effective for taxane-resistant tumors. Few after effects like sensory neuropathy and fatigue are correlated with this drug. Another antimitotic agent, known as lalulimide, is derived from marine sponges and has a complicated structure. It attaches to a particular location on alpha-tubulin and has the property of microtubule stabilizing. One more drug which is extracted is fluoroside A from marine sponges, which has a similar structure to apopolones. It binds on alpha-tubulin to the lalulimide binding site. Dictostate and discodermalide are naturally obtained from marine whites that use these microtubules to be a fundamental system of defense. This includes the assembly of tubulin, enhanced stability of the microtubules, hypernucleation, and reduced depolymerization. Microtubule destabilizing drugs. The important class of antimitotic drugs is the depolymerizers, which include drugs that attach to the tubulin domain of Vinca or colchicine. They destabilize the structure of microtubules at elevated concentrations. They perform their functions by disturbing the microtubule dynamics and by reducing polymerization. Eventually, apoptosis will occur by the block dividing cells in mitosis. These Vinca domain binders may be classified as Vinca site binders and protein site binders by agents like Vinca alkaloids, interact with the attachment of vinblastine, and have a destination location of the Vinca domain on tubulin. By extracting them from crops, microbes, and marine organisms, Vinca alkaloids can be obtained in essence. The Vinca alkaloids are basically a group of antimitotic compounds extracted from the periwinkle plant, Catharanthus roseus, which function by attaching tubulin to the beta subunit near the GTP binding site and by restricting mitosis from metaphase. Vinblastine and vincristine are major examples of first-generation Vinca alkaloids. They change the conformation of dimers, inhibit tubulin-dependent GPT hydrolysis, and change the GTP exchange near the hydrolyzable GTP site. Vincas bind to the plus ends of microtubules when present in low concentration, limiting dynamics and eventually arresting mitosis. Vincas are also referred to as and poisons because of this nature. Vindicine, phenoralbine, and vinfluline are semi-synthetic alkaloids of vinca. Another type of drug, maytansinoids, for example, maytansine, obtained from the plant, Maytanis ovitis, is an anson macrolide. Maytansine basically restrains the aggregates formed by vinblastine leading to their dissolution. Vinca alkaloids, mainly vincristine, obstruct the maytansine's binding site. Rhizoxin, which is derived from a fungus, Rhizopus chinensis, is a macrocyclic lactone that has an meiotic arrest of cells and further disintegration of MTs. Delastatin 10 non-competitively limits tubulin-independent GTP hydrolysis binding of vincristine and tubulin-dependent restraints along with nucleotide exchange. The attachment of fomoxin A and rhizoxin to tubulin is also inhibited by delastatin 10. Lactone polyethers like spongistatin are obtained from marine sponges such as Phorasphyllus spinospiraliferas. They are cytotoxic in nature and potently restrict the binding of delastatin 10 and vinblastine. Spongistatins also result in the cell's meiotic arrest and dissolution of microtubule assembly. Colchicine domain binders. The colchicine binding location is located at the junction of the alpha and beta tubulin monomer directly in the center of the tubulin dimer. It lies in the filament's hollow lumen. Subsequently, binding at the colchicine site results in a conformation change that causes an intradimer distortion. This conformational modification allows the colchicine tubulin complex to be involved within the filament of the microtubule. Compound colchicine was first acquired from saffron, Colchicum autumnale. It is one of the earliest targeting agents of microtubules. Colchicine played a significant role in determining microtubule characteristics in its tubulin subunits as it binds potently to tubulin, known as a high-propensity colchicine binding protein. Almost 7 beta tubulin and 6 alpha tubulin isotypes have been investigated in mammals. Colchicine molecule has 3 rings, A, B, and C. Colchicine's B ring is critical for its restrictive tubulin binding feature. The analog colchicine missing this ring 2-methoxy 5-2,3,4,5-trimethoxyphenyl attaches simultaneously to tubulin. Microtubule depolymerizers, for instance, colchicine and other drugs which attaches to the colchicine site have been exploited as potential cancer therapeutic agents. Combretistatins are also antimitotic agents that are acquired from Combretum cafrum, a South African tree. The most prospective members of this class are Combretistatins A4 and Combretistatins A2. In addition, significant antitumor activity. This macrolide constrains tubulin's binding because it partakes a common binding site with maytansine. In addition, at elevated levels, rhizoxin does not form aggregates. Furthermore, ansamidosins are extracted from gram-positive nocardia species, and also antiproliferative activity. Furthermore, ansamidosins are extracted from gram-positive nocardia species, and also antiproliferative activity is structurally similar to anson macrolide. Fomoxin A and mucilaxins are basically comparison peptides of contagious starting points that induce meiotic capture at tiny molar centers and facilitate meiotic arrest at very less micromolar concentrations. Both are derived from the fungus Phomopsis leptosomoformis and mucilaginum adherens, respectively. As maytansine and mucilaxin A, fomoxin obstructs tubulin polymerization and precludes tubulin-supported nucleotide hydrolysis. The powerful mucilaxins restrict MT gathering whereas usiloxin balances tubulin configuration. Various studies demonstrate that fomoxin form the spiral tubulin aggregates. The powerful usiloxins restrict MT gathering whereas usiloxin balances tubulin configuration. Various studies demonstrate that fomoxin form the spiral tubulin aggregates. Other instances of microtubules destabilizing anticancer agents are halochandrins and halostatins of marine descent. Halochandrin B is basically a polyether of lactone obtained from the Halochandria opidae marine sponge. Even more, halostatin is acquired from sponge axonella species. Halostatin impacts tubulin formation in vivo and restricts GTP hydrolysis and nucleotide swap subordinated to tubulin. Indeed, MT's polymerization is inhibited by various marine and microbial compounds. Since the touching places of these organic compounds on tubulin partially overlap with vinca alkaloids, and several are peptides, modified peptides, cyclic peptides, or DEPC peptides, they are acknowledged as inhabiting the vinca domain's peptide site. Compounds such as delastatins, cryptophycins, and spongistatins are the well-recognized anticancer agents which bind to the vinca domain's peptide site. Delastatins are the natural peptides extracted from a species of the sea hare, Dolabella auricularia, and they are byproducts of a pseudopeptide with 4 unique amino acids. Delastatin is the most compelling antimitotic substance resulting. Proteins interacting with microtubule dynamics are significant for the intracellular functions of the microtubule cytoskeleton and are subsequently exceedingly directed. Proteins that control microtubule dynamics lie into 2 major categories: proteins stabilizing the microtubules and microtubules destabilizing proteins. MTs are actually stabilized in many ways: I by precluding catastrophe, growth perseverance of MT, and or lengthened contact with target sites; E by preventing it depolymerizing MT and hence reducing the shortening phase, or E by reducing the speed of shrinkage. Few proteins alter the tubulin heterodimers conformation and bring in kinks that favor MT depolymerization and therefore give rise to catastrophes. For instance, Stapman and kinesin 13s. Both proteins decrease procurable subunits and control the effectiveness of polymerization. Furthermore, collapsin response mediator protein CRMP2 enhances MT growth by encouraging tubulin dimers addition onto the microtubule plus ends. Other proteins that interact with assembled MTs are those that bundle MTs MAPC, those that stabilize MTs tau, and those that keep MTs dynamics MAPB. One important signaling pathway that modulates MT dynamics includes GSK3 beta, a kinase mostly active under basal growth conditions, increasing the growth and dynamics of microtubules. In vitro, MCAK proteins attach to both minus and plus ends and reveal the highest inclination for curved protofilaments. Two proteins, Opus 18 and XKCM1, act as powerful microtubule destabilizers, and these proteins also bind tubulin dimers in vitro. Xenopus microtubule-associated protein 215, XMAP215, also enhances MT assembly via binding to tubulin dimer. Microtubule-associated proteins, MAPS. Microtubule-associated proteins, MAPS, make up at least 10-15% of the isolated mass of microtubules. These MAPS bind to the microtubular wall at constant intervals. To inhibiting the colchicine binding site in tubulin polymerization, they are referred to as antineoplastic agents by binding to tubulin, inhibiting the cycle of cells at mitosis and ultimately leading to apoptosis. Combredastatine also inhibits angiogenesis, used to target tumor vasculature. High levels of combredastatins are adequate to inhibit the assembly of microtubules via tubulin-dependent GTP hydrolysis. In addition, 2-methoxyestradiol, 2-ME, is a naturally occurring mammalian derivative of beta-estradiol estrogen hormone. It has antitumorigenic and anti-angiogenic properties and leads to apoptosis in endothelial cells. 2-ME obstructs the tubulin assembly because of its innervated competitive inhibition with colchicine binding. After assembling into a polymer instead of forming a tubulin 2-ME complex, 2-ME attaches to tubulin and participates in polymerization. For the treatment of gout, constipation, syphilis, tuberculosis, rheumatism, liver cirrhosis, and cancer, another compound, podophyllitoxin, and its analogs are separated from the plant Podophyllum peltatum. Podophyllitoxin inhibits competitive inhibition of colchicine binding to the colchicine binding domain and shows a faster and more reversible tubulin binding relative to colchicine. Podophyllitoxin reveals tubulin-dependent GTP hydrolysis. Albeit podophyllitoxin and colchicine attach on beta tubulin to the comparable site, there is no absolute overlap in the binding site, and both have different binding patterns. Another class of binding agent for the colchicine domain is chalcones. Chalcones, Pterogramma, and several other plant products, for example, ferns, Physis melanogenosum, Hyperoduncum, were acquired from grasses. Chalcones are 300 times more efficient than colchicine in cell cycle arrest and have strong cytotoxic characteristics. Chalcones like trans 1,2, 5-dimethoxy-3,4-dimethylamino, phenyl -2-methyl-2-propane-1,1, MDL, are powerful antimitotic agents. They bind reversibly to the beta tubulin colchicine site and inhibit microtubules assembly, letting the interaction with filaments and other cellular structures and enhance MT stability. MAPS may be classified into two groups based on their function. One group balances microtubules in a stabilizing MAP structure consisting of two fields, a basic binding domain of microtubules and an acid projection domain. The projection domain appears under the electron microscope as a filamentous arm extending from the microtubule wall. This arm interacts with intermediate filaments, membranes, and other microtubules. The binding domain of microtubules includes many repetitions of a preserved, positively charged sequence of 4 amino acid residues that interact with tubulin's negatively charged C-terminal. This binding nullifies the charge revulsion between tubulin subunits, therefore stabilizing the polymer structure. Other stabilizing MAPs are tau, MAP4, MAP2, and CLIP170. MAP4 is the most prevalent of a considerable number of MAPs and is found in non-neuronal cells. During mitosis, it modulates the stability of microtubules and CLIP170 links microtubules to chromosomes. MAP2 is discovered distinctly in dendrites, where fibrous cross bridges are framed between microtubules and microtubules are connected to intermediate filaments. Another MAP, tau, is found in axons and dendrites and is involved in axonal elongation. Tau's quality of cross-interface microtubules into thick bundles enhances axonal microtubules stability. Deletion of the encoding genes tau and MAPIB encourage more terrible axonal dysgenesis and lethality phenotypes. In addition, tau's modified polymerization in filaments is associated with neurodegenerative diseases such as human dementia in patients with Alzheimer's disease. Also, tau and MAP4 stabilize microtubules, and the attachment of MAPs can regulate the length of the microtubule. Phosphorylated MAPs cannot bind to microtubules, hence modulate microtubule disassembly. MAP kinase, a significant enzyme for phosphorylated MAPs, is involved in several signal transduction pathways, demonstrating that MAPs are targeted in different extracellular signals. MAPs, particularly MAP4, are phosphorylated by cyclin-dependent kinase (CDK) that plays a significant role during the cell cycle in controlling the different protein operations. MAP's second group destabilizes microtubules in different cell kinds. One member of this group is catenin, which displaces integral cytosolic microtubules by breaking the internal bonds among tubulin subunits in the wall of the microtubule with the help of ATP. The frequency of quick microtubular disassembly in the meiotic spindle is increased by another protein called Opus 18 or Stathmin, which carries out its tasks by binding the tubulin dimer to decrease the dimer pool, procurable for polymerization. Opus 18 is inactivated by phosphorylation and obstructs its destabilizing effect. Other MAPs that interact with the microtubules and are microtubule regulatory kinesins, microtubule depolymerases and polymerases that help in determining the microtubule growth or shrinkage. Figure 4.44 microtubule interacting proteins. A portion of tau, a MAP binds at MT length, other part of tau escalates away from the MT. Controlling spacing of MT. B plus tip proteins, for example, CLIP170, bind near the plus ends of MTs and stabilize them. C catastrophins, for instance, MCAK, a kinesin family protein, destabilize MTs. Plus end tracking proteins plus tips polarity of microtubules is evident in the distinctive, unique properties of their ends. The quickly growing end of the microtubule with beta subunits of tubulin revealed is called the plus end, and the contrary end is called the minus end. The minus end is mostly stabilized and present closer to the center, near to the centrosome, whereas the plus end is very dynamic and pointed towards the boundary. Microtubule plus ends dynamics are controlled via several proteins affiliating with tubulins. The plus tip interacting proteins, also referred to as plus end tracking proteins, plus tips, are a unique class of proteins associated with microtubule. They control the various structure and functions of microtubules and are chiefly present on the growing ends of microtubules. Some examples of plus tips are microtubule polymerase XMAP215, Xenopus microtubule-associated protein, 215,000 Da. This protein belongs to the kinesin family and the accelerated polymerization or depolymerization of microtubules. Several plus tips are involved in MT stabilization by joining them to the cell cortex. Clasped cytoplasmic linker protein-associated proteins and ACF7 ATP-dependent chromatin assembly and remodeling factor 7 enact as microtubule salvation and cell cortex stabilizers, although APC adenomatous polyposis coli decreases catastrophes at MTs and cell projections. Clipped cytoplasmic linker proteins behave like a cytosolic rescue factors by combining with growing MTs. In addition, under the cap, GDP-bound tubulin dimers have GTPase activities. This cap includes proteins that are end-binding EB. Basically, EB proteins are the evolutionarily preserved proteins that track the growing microtubule end autonomously. They control the dynamics of microtubules and are also responsible for selecting the most different plus end tracking proteins to the microtubule's growing end. EB1, EB3, and fission yeast homolog mal 3 permit relentless MT growth in cells by hindering catastrophes. EB proteins interface with the tips of developing MTs. The most powerful protein that appeared to build MT polymerization phase is XMAP215, a broadly expressed and exceedingly conserved protein present in every single eukaryotic cell. In vitro, XMAP215 increase the assembly, and both STU2 as well as XMAP215 wrap around a tubulin heterodimer and encourage its acquisition into a developing plus end without the inclusion of ATP. Hence, XMAP215 enhances both growth and shrinkage and controls phase transitions in vitro and in cell extracts. Binding to EBs is crucial for plus tips to amass at the plus end of MT for plus end tracking. Motor proteins. Motor proteins are the major components of the class of molecular motors that can travel along the cytosol of animal cells. They change chemical energy into mechanical work with the help of ATP. Hydrolysis. Motor proteins generate force within the cytoskeletal polymers network that further performs such forces to figure out the structure for each cell and eventually, the tissue architecture and the whole organisms. Myosin, kinesins and dyneins are members of 3 main motor protein families and conduct most eukaryotic movements concerning the movement of microtubules and microfilaments. Myosin, kinesin and Ross triphosphatases GTPases have a prevalent ancient source, but dynein is a part of the triple triphosphatase adenosine ATPase family, even though none of these motor proteins was discovered in prokaryotes. Gradually, eukaryote gene duplication and variation generated many genes for myosin, dynein, and kinesin, each with distinctive features encoding proteins. There are genes for 5 myosins, 6 kinesins, and 1 dynein in the budding yeast genome. There are 2 components of a motor protein: a motor area that utilizes ATP hydrolysis to create motion and a tail that pushes motors to blend or attach particular products. The heads in the three houses are very distinct from the motor area, allowing the unique functions of each motor isoform. All motor proteins are enzymes that transform chemical energy stored in ATP into the molecular movement to produce a force on the polymer of the cytoskeleton. The polymer passes when the motor is attached. The motor and other attached goods move when the polymer is fastened. If both are anchored, the pressure transiently extends elastic components in the molecules, but nothing moves but dissipates the energy as heat. All these options are basically exploited by the cells. Myosins are the only motors which use actin filaments as trails. Myosin superfamily participants come from a prevalent ancestor and take part in a motor unit recognized as a myosin head that forces on action filaments. One or two heads are connected to multiple kinds of tails that are applematized for different reasons, including binding membranes, polymerizing filaments, and multiple cargo affiliations. Motor kinesin and dynein relatives use ATP hydrolysis power to replace membrane-bound organelles, vesicles, chromosomes, and cargo along microtubules. Dynein also assists with eukaryotic cilia and flagella bending movements. Dyneins migrate toward the minus end of microtubules, along with some goods. The dynein family of motor proteins includes two types, axonal dynein and cytoplasmic dynein. The cytoplasmic dynein includes two heavy chains, two intermediate chains, two intermediate light chains, and several light chains that combine with microtubules. Cytoplasmic dynein shifts to the minus end of MTs and is associated with a complex of proteins called dynactin. The dynactin complex aids in attaching cytoplasmic dynein with cargo, such as membranous vesicles. It carries along MTs by attaching to proteins like spectrins attachment with the membrane of its cargo. Most kinesins migrate to the plus end, but a few members of the kinesin family are myosin-driven motors and some others promote the disassembly of microtubules. Microtubule motors have heads and tails that contact cargo with at-pace activity. As a molecular motor, kinesin is effective in transforming the ATP hydrolysis energy into beneficial work, 60–70% efficient. Kinesins carry a diverse array of cargo, such as chromosomes and organelles, along microtubules. The first kinesin was initially recognized in the cytoplasm of squid giant axons, comprised of three parts: a globular head region that interacts with microtubules and involves ATP hydrolysis, a coiled helical region, and a region of light chains that attaches kinesin to other proteins or organelles. Kinesins move along MTs in a few steps. One of the two globular heads moves forward to get attached to a beta tubulin subunit, succeeded by the detachment of the trailing globular head, which makes an attachment to a new region of MT. This movement is coupled with ATP hydrolysis bound at the unique sites within the heads. This results in the movement of kinesin toward the plus end of an MT in a manner dependent on ATP. Kinesin 1 is a motor protein which moves cargo, for instance, organelle, incessantly in the heading of the plus end of a microtubule. A molecule of single kinesin 1 can cover long distances before detaching from an MT. For this, it liberates the bound ATP and obtains a new molecule of ATP, permitting a cycle to repeat. A molecule of single kinesin can move up to 1 millimeter, a considerable distance for much of its small size. Kinesin 1 shifts membrane vesicles away from the centrosome and along neuron axons to the plus end of microtubules. Kinesin 2 is the motor for particle movement in cilia and flagella towards the axoneme tip intraflagellar transport. It carries in the cytoplasm a different type of cargo over long distances. Kinesin 4 motors attach both DNA and microtubules called chromo-inclusive kinesins. They move cargo in the neurons along axons. Motors based on dynein microtubules are AAA passes, consisting of 6 at-pace domains. During interactions with microtubules that produce force, the AAA1 domain of dynein binds and hydrolyzes ATP. When AAA1 is nucleotide-free, dynein binds closely between the subunits of alpha and beta tubulin to a microtubule with the stalk facing towards the polymer's minus end. Figure 4.45 Movement of kinesin. Of the fundamental structure of various kinesin family members. Bead kinesins walk along microtubules. One globular head of kinesin front foot detaches from a beta tubulin subunit by hydrolyzing ATP and moves forward. The back foot gets released by ATP hydrolysis and then moves to the front. Microtubule dynamics and motor proteins in mitosis. The orderly sequence of events during which a cell duplicates its contents and gives rise to two daughter cells is termed the cell cycle. Replication of the genome and its proper distribution into daughter cells during division are the fundamental events of the cell cycle. Typically, a eukaryotic cell cycle consists of 4 sequential phases, namely G1, G for gap, S for synthesis, G2, and M phase, M for mitosis. G1, S, and G2 are collectively termed as the interphase, during which DNA duplication and synthesis of factors required for growth and division occurs. The cell cycle culminates with mitosis, characterized by progressive condensation of the chromatin into compact chromosomes and chromosome segregation followed by both nuclear and cytoplasmic division. Mitosis is categorized into 5 distinct stages, namely prophase, prometaphase, metaphase, anaphase, and telophase. Each of the phases is characterized by alterations in the chromosome structure. The primary goal of mitosis is the segregation of chromosomes with high fidelity. Errors during chromosome separation result in aneuploidy, thereby leading to genomic instability and cancer. A highly coordinated mechanism that enables an error-free meiotic progression is carried out by microtubule motor proteins and microtubule assembly dynamics, which will be discussed at length in this chapter. The meiotic apparatus. In eukaryotic cells, a macromolecular machine termed as a meiotic apparatus, composed primarily of dynamic microtubule polymers and formed between the opposite poles of a meiotic cell governs chromosome segregation. The first detailed drawing of the meiotic spindle in early Ascaris embryos by Theodore Boveri in 1901 shows chromosomes attached to a bipolar network of fibers, Fig 4.46. He showed that spindle fibers emanate from structures at each pole that we now identify as centrosomes, and he also noted that each centrosome contains 2 small rod-like bodies, which are recognized as centrioles. The composition of the meiotic spindle was unknown until the discovery of tubulin in the 1960s. Visualization of the spindles by electron microscopy helped determine that spindle fibers are bipolar arrays of microtubules composed of tubulin subunits. The major components of the meiotic spindle is microtubules, microtubule-associated proteins, MAPs, and microtubule organizing center, MTOC. Figure 4.46 Different stages of mitosis. Mitosis is divided into 5 distinct stages, namely prophase, prometaphase, metaphase, anaphase, and telophase. Prophase is characterized by a breakdown of the nuclear envelope and chromosome condensation. The centrioles divide and move apart. During prometaphase, the centrioles and asters migrate towards the opposite poles. The nuclear membrane is completely disintegrated, and the spindle begins to interact with the chromosomes. The chromosomes, each with 2 sister chromatids, align at the center of the cell during metaphase. Anaphase is marked by a separation of the sister chromatids and poleward movement to the opposite ends of the spindle. Following this, telophase sets in where the cell membrane constricts at the center to divide the cell into 2 daughter cells. Nuclear membrane forms around each of the 2 sets of chromosomes. The chromosomes begin to decondense and become diffused. Cytokinesis defines the successful completion of mitosis, whereby the cell is physically separated into 2 identical daughter cells. Microtubules are the fundamental structural and functional constituent of the meiotic apparatus, and their intrinsic polarity and dynamicity are critical for spindle morphogenesis. These microtubule filaments grow and shrink by the respective addition and loss of tubulin dimers at their ends. They are arranged in a bipolar array with their minus ends at the poles and plus ends towards the spindle. During prophase, microtubule shrinkage and lower levels of growth disrupt the interphase microtubules, promoting interaction between the dynamic plus ends and chromosomes. The chromosomes begin to align themselves at the spindle equator, and during metaphase, all the chromosomes are present at the spindle equator, also known as the metaphase plate. At each half of a uniformly oriented spindle, a single centrosome organizes 3 sets of microtubules during this phase. Firstly, kinetochore microtubules attach to the chromosomes at specific points of attachment on the paired chromatids called the kinetochore, with their minus end proximal to the centrosome. Secondly, the interpolar microtubules extend towards the central spindle, forming an overlapping antiparallel network with polar microtubules at the opposite poles. The third subset includes the astral microtubules, which radiate from the poles towards the cell cortex away from the spindle and are involved in positioning the spindle in the cell. The dynamic polarity and antiparallel organization of the microtubules are instrumental for proper meiotic spindle assembly. The end-on interactions between spindle fibers and the kinetochore of sister chromatids and interactions between overlapping ends of interpolar microtubules form the bipolar spindle architecture. In anaphase, cohesion between the sister chromatids is lost, allowing them to separate. The poleward movement of the new daughter chromosomes leads to a shortening of the kinetochore microtubules. Telophase marks the completion of mitosis, where further separation of the chromosomes towards the opposite poles takes place. The chromosomes undergo decondensation, and the nuclear envelope reappears around the daughter nuclei. The cells then progress towards cytokinesis, where the cytoplasmic division of the parent cell occurs. The centrosomes are the fundamental microtubule organizing center, MTOC, in mammalian cells. They function to trigger the polymerization of microtubules and serve as an anchor to the astral microtubule subset, which extends towards the cell periphery. The centrosome consists of a pair of centrioles surrounded by paracentriolar material, PCM, where nucleation occurs, thereby producing polarized microtubule filaments. Gamma-tubulin and paracentrin are the major components of PCM, which assemble to form a multi-subunit gamma-tubulin ring complex gamma-turk. The organization and function of centrosomes undergo dramatic changes like replication and maturation during cell cycle progression. After maturation, the centrosomes organize microtubules into a highly complex and robust meiotic spindle apparatus for chromosome segregation during mitosis. Figure 4.47 Meiotic spindle architecture during metaphase. The microtubules emanate from the centrosomes with their -ends tethered at the spindle poles. Some microtubules attach with the kinetochore of paired chromatids, known as spindle microtubules or kinetochore fibers. Some form an overlapping antiparallel array termed as interpolar microtubules, and another subset extends from the poles towards the cell cortex called astral microtubules. -end directed motor proteins like dynein exert pulling forces on the astral microtubules, which help in proper spindle positioning. Another group of motor proteins called chromokinesins promotes the +end directed movement. The kinetochore. The dynamic movement of microtubules during mitosis is driven and regulated by a large, evolutionary conserved protein complex called the kinetochore, which serves as the interface between the chromosomes and the spindle microtubules. The kinetochore is assembled on a specialized region of the chromosome, recognized as a centromere, and is subdivided into an inner and outer region. The inner region of the kinetochore exists as a heterochromatin domain during the cell cycle, and the outer layer is a proteinaceous structure containing several components that function solely during mitosis to enable attachment of chromosomes to spindle microtubules, generation of force for chromosome movement along the kinetochore microtubules, and ensuring chromosome segregation and translocation to the respective spindle poles. Fig 4.47. RNA interference (RNAi) studies in C. elegans, Drosophila, and human cells and gene knockouts in mice and cultured chicken cells have helped gain insight into kinetochore assembly. The key protein in determining the site of a functional kinetochore is the histone H3 isoform centromeric protein, CNPA. Additionally, CNPA is required for the recruitment of kinetochore inner core proteins like CNPC, CNPH, and CNPI Mis6. Studies in C. elegans revealed that CNPA and CNPC govern the localization of KNL1 and KNL3 proteins, which are essential for the assembly of multiple components of the outer kinetochore and the formation of a functional kinetochore microtubule attachment. The KNL proteins further govern the recruitment of several outer plate proteins of the NDC80 complex, Mis12, CNPF, and the checkpoint protein BUB1. Recently, in 2004, a KNL1 homolog AF15Q14 was identified in human cells, suggesting that the CNPA-mediated kinetochore assembly pathway is conserved. A bunch of other kinetochore proteins that depend on the CNPA-mediated pathway for targeting include polo-like kinase PLK, the microtubule motor dynein, the kinesin motor CNPD, meiotic kinesin MCAK, the spindle checkpoint proteins MPS1, BUBR1, MAD1, MAD2, and the non-motor microtubule-associated proteins MAPS clasps. These proteins work collectively to facilitate the attachment of the kinetochore to a microtubule end and eventually translocate the chromosomes to opposite ends of the cell during mitosis. Based on their localization during the cell cycle, these proteins are categorized into three groups. CNPA, CNPC, CNPH, and CNPI proteins are bound to kinetochore-associated chromatin and their levels remain constant throughout the cell cycle. They are termed constitutive components. The concentration of other kinetochore proteins also be grouped vary during mitosis and depending on their turnover rate. They are grouped into stable slow turnover and dynamic rapid turnover. NDC80 complex proteins, KNL, KBP proteins, Mis proteins, and CNPF that constitute the core inner and outer kinetochore regions are examples of stable kinetochore proteins. Lastly, the dynamic kinetochore components that change in concentration during mitosis include the microtubule motor CNPD and dynein and the spindle checkpoint proteins e.g. MAD1, MAD2, BUBR1, and CDC20. A high concentration of these proteins is present at kinetochores in the absence of microtubules, and their concentration gradually drops during microtubule attachment. By metaphase, CNPD, BUB3, and BUB1 levels are decreased 3 to 4-fold relative to those at unattached kinetochores, and dynein, dynactin, MAD1, MAD2, and BUBR1 levels also undergo a dramatic reduction. Several groups of researchers have measured the dynamics of protein turnover at kinetochores by fluorescent recovery after photobleaching, FRAP, of green fluorescent protein GFP fusion proteins expressed in cells. CNPA, CNPI, CNPH, NUC2, MAD1, and BUB1 are stable components that turn over very slowly over 10 minutes. On the other hand, the spindle checkpoint proteins, namely 50% of MAD2 and most of BUB3, BUBR1, and CDC20, are very dynamic components with high turnover rates of less than 30 seconds. The spindle checkpoint impedes errors in chromosome segregation by inhibiting anaphase onset until all chromosomes have aligned at the spindle equator through the attachment of their sister kinetochores to microtubules from opposite spindle poles. The figure shows the localization of fluorescent MAD2 in a living meiotic PTK1 cell. Fluorescent MAD2 localized to kinetochores in prophase A and early prometaphase B and to spindle poles in prometaphase C. Fluorescent X MAD2 diminishes at kinetochores in prometaphase B and C and at kinetochores and spindle poles in early metaphase D. The cells enter anaphase F once MAD2 has been depleted from all kinetochores at late metaphase E. This fast turnover of the spindle checkpoint proteins at the kinetochore thus facilitates the ability of unattached kinetochores to inhibit activation of the anaphase promoting complex. Therefore, the structural plasticity of the kinetochore components promotes accurate chromosome segregation in yeasts as well as vertebrates. The centromere. The centromere is a discrete locus on each chromosome that drives the distribution of one copy of each chromosome to each of the daughter cells during cell division. The centromere was first described by German biologist Walter Flemming in the 1880s as the primary constriction of the chromosome. After several genetic and biochemical studies, scientists have now determined that the centromere is a region of specialized chromatin found within each constricted chromosome that provides the platform for kinetochore assembly and serves as a site for sister chromatid attachment. The centromere is visualized as the most constricted region of a condensed meiotic chromosome and is located at different positions, characteristic of each chromosome. Based on the location of the centromere, chromosomes are classified into 4 types, namely metacentric, submetacentric, acrocentric, and telecentric. Thus, the position of the centromere provides a useful landmark for categorizing chromosomes into karyotype groups and for establishing a standardized nomenclature for mapping positions of genes on the chromosomes. Centromeres in budding yeast are formed upon DNA of a defined sequence, called point centromere, whereas centrosomes in higher eukaryotes built on tandem repeats of highly repetitive elements are called regional centromeres. Despite the large divergence of centromeric DNA across organisms, the centromeric-associated proteins, CNPs, are highly conserved. Among the CNPs, CNPA and C are the most extensively studied centromere-associated proteins. CNPA acts as a histone H3 variant involved in the assembly of a functional kinetochore. CNPA was shown to co-purify with nucleosome core particles 33, which indicated that it probably forms a complex with the core histones. CNPA is essential for centromere and kinetochore formation in all organisms. CNPA is recruited to centromeric chromatin in 2 steps. During the S phase, when DNA replication occurs and nucleosomes are recruited to the duplicated DNA, CNPA is deposited onto the new chromatid. The second step involves the deposition of newly synthesized CNPA after mitosis in early G1. This process requires CNPA-specific factors, including Mis18-KNL2, like a group of proteins in C. elegans, and vertebrates as well as CNPC. CNPC is a DNA-binding protein that associates with the inner kinetochore plate, Ref34, and is pivotal for proper meiotic progression and chromosome segregation. CNPC depletion at the centromere resulted in aberrant kinetochore formation. Localization of CNPC to centromeric chromatin required the presence of CNPA. CNPC interacts with 2 different groups of proteins that serve distinct functions, the Mis12 complex 41, 42, which is part of the KMN network, which comprises the KNL1 complex, Mis12 complex, and NDC80 complex of the outer kinetochore that is also needed for recruitment of checkpoint proteins, and to other constitutive centromere-associated network CCAN components, such as CNPH, CNPI, CNPK, and CNPT. Additionally, CNPC binds distinct CNPA domains to regulate the assembly of other centromere and kinetochore proteins. The CNPT-CNPW subcomplex, consisting of CNPH, CNPI, and CNPK functions upstream of CNPH and are recruited to the centromere by CNPC. These proteins are implicated in kinetochore function in vertebrates and are involved in the recruitment of CNPA and other centromere complexes. The CNPS subcomplex, comprising CNPS and CNPX, is required for the stable formation of the outer kinetochore. The CNPO subcomplex, made up of CNPO, CNPP, CNPQ, and CNPR and CNP50, is needed to prevent premature separation upon spindle damage. Thus, the centromere is an essential site on every eukaryotic chromosome, and errors can result in a wide range of diseases, including cancer and aneuploidy-related disorders. Future work will incorporate the contributions to develop a comprehensive model of centromere kinetochore attachment and the nature of the centromeric and paracentromeric chromatin. Kinesin and dynein. Axonal transport. Axonal transport is fundamental for neuronal cell function and morphogenesis. Defects in the axonal transport machinery are responsible for developmental and neurodegenerative diseases. The neuron consists of the cell body, dendrites, and a single axon. Anterograde axonal transport involves the movement of newly synthesized proteins and lipids from the cell body to the axon. A complementary mechanism that usually functions to translocate misfolded or aggregated proteins from the axon to the soma is termed retrograde axonal transport. Experiments with drugs that disrupt the cellular cytoskeleton reveal that microtubules play a central role in axonal transport. Microtubules are polarized tubulin polymers with fast-growing plus ends oriented outwards and stable minus ends towards the cell body. In the axon, parallel microtubules form a unipolar array with their plus ends towards the terminal, while in dendrites, microtubule organization is more complex, with microtubules organized in arrays with mixed polarity. Experimental evidence showing axonal transport. The bidirectional axonal transport was discovered by pulse chase experiments, where the dorsal root ganglia cells near the spinal cord are microinjected with radioactive precursors. The movement of these labeled compounds was tracked along their axons, which unraveled that axonal transport occurs in two directions, namely anterograde and retrograde transport, Fig 4.48. Membrane-bound vesicles migrated outward from the cell body at the highest speed of about 400 mm per day or 1 micrometer per second. Organelles moved along the axon at an intermediate velocity of about 250 mm per day or 3 micrometers per second. Cytoskeletal proteins slowed the slowest speed of transport. Development of live cell imaging techniques that enable visualization studies of organelle motility led to the discovery of motor proteins involved in axonal transport. Figure 4.48 Injection of radiolabeled amino acids into a dorsal ganglion of mice. Mice are sacrificed at different times after injection, and the sciatic nerve is dissected and cut into 5 mm segments. Initially, the cell bodies in the ganglion integrate the labeled amino acids into proteins, which then are transported along the axon. Soon after injection, labeled proteins are found only in segments closest to the cell body. With increasing duration of chase, labeled protein is detected distal to the cell body bottom. The red, blue, and purple dots represent groups of proteins that are transported down the axon at different rates, red most rapidly, purple least rapidly. The amount of radiolabeled protein in each fragment is measured, and then the various proteins are resolved by gel electrophoresis. Axonal transport is visualized in vitro by video micrograph studies of the cytoplasm extruded from a squid axon. These studies show that ATP is necessary for the movement of vesicles along microtubules in this cell-free setup. Electron imaging studies conclude that organelles move along individual microtubules. These experiments laid the foundation for the discovery of motor proteins pivotal for axonal transport. Kinesin, A plus N directed motor protein. Kinesin is a primary motor protein involved in the anterograde transport of cargos, like membrane-bound vesicles, and protein complexes towards the microtubule plus ends. This plus N-directed 380 kD motor protein consists of two 120 kDa kinesin heavy chains, KHCs, that possess the motor activity and two 64 kD kinesin light chains, KLCs, that enable cargo binding. Experimental evidence to study the kinesin-dependent vesicle movement in vitro motility assays based on video-enabled microscopy was instrumental in determining the nature of kinesin. Axoplasm from the squid axon was separated into soluble and organelle fractions that lacked microtubules. Microtubules were constituted from squid tubulin and stabilized using Taxol. These fractions were then combined on a cover slip chamber to assess their motility. Purified vesicles coated with the soluble kinesin bind to the purified microtubules, and in the presence of ATP and move along the plus end of the microtubules. A similar observation was reported when latex beads were coated with kinesin, and their motility was recorded. The soluble fraction alone adsorbed onto the glass cover slip, causing microtubules to glide across the surface, with their minus ends in the lead. This experiment concluded that kinesin is a plus N-directed microtubule motor protein responsible for anterograde cargo transport. Kinesin 1 was the only motor protein known to migrate towards the plus ends of microtubules. Several kinesin-related proteins that belong to the kinesin superfamily were discovered in the early 1990s, after the first sequence of a gene encoding a kinesin 1 heavy chain, Drosophila melanogaster KHC, was deposited into the public databases. The well-characterized kinesin superfamily proteins and their biological functions in organelle transport are discussed below. Figure 4.49 Model of kinesin-catalyzed anterograde transport. A kinesin motor-dependent transport of vesicles along microtubules. The kinesin molecules, attached to unidentified receptors on the vesicle surface, transport the vesicles from the minus to the plus end of a stationary microtubule. The kinesin molecules bound to the glass surface move toward the plus end of the microtubule. Because the kinesin molecules are immobilized onto the cover slip, the sliding force is transmitted to the microtubule, which then moves in the direction of its minus end. Dynein, A minus N-directed motor protein. Dynein is a multi-subunit large motor protein complex that governs minus N-directed intracellular transport. Three major types of dynein exist in mammalian cells: axonal dynein, cytoplasmic dynein 1, and cytoplasmic dynein 2. Axonal dynein is present in cilia and flagella and is mainly involved in coordinating the beating motion of both cilia and flagella. Cytoplasmic dynein 2 is also found in cilia and flagella and is involved in cargo protein transportation within cilia and flagella structures. Cytoplasmic dynein 1, commonly known as cytoplasmic dynein, is a major form of cytoplasmic dynein found in mammalian cells and is ubiquitously expressed. Cytoplasmic dynein 1 is involved in minus N-directed transportation of various cargo proteins, organelle positioning, cell migration, and meiotic spindle assembly. Cellular functions of dynein. The primary cellular function of cytoplasmic dynein is retrograde axonal transport and movement of protein and RNA complexes, Golgi apparatus, lysosomes, and endosomal vesicles. Cytoplasmic dynein 1 is also associated with kinetochores and has multiple roles during mitosis. Dynein-dynactin complexes are shown to localize at the nuclear envelope immediately before the onset of nuclear envelope breakdown, NEBD, highlighting the importance of dynein for proper NEBD. Dynein is also necessary for the migration of duplicated centrosomes for bipolar spindle assembly. Moreover, perturbation of dynein function either through RNAi-mediated depletion or microinjection of inhibiting antibodies impaired centrosome separation, resulting in aberrant bipolar spindle formation. These observations suggest that cytoplasmic dynein is involved in centrosome separation during the onset of mitosis.